Antigen-presenting cells, or APCs, are specialized immune cells that play a crucial role in our immune response. They process antigens and then present fragments of these antigens on their surface, bound to class II MHC molecules. This presentation is essential because it allows CD4-positive T helper cells to recognize and respond to the antigens. APCs are central to initiating and regulating our adaptive immune responses, which include both humoral immunity, mediated by B cells, and cell-mediated immunity, driven by T cells. They not only present antigens, but also provide vital co-stimulatory signals that are necessary for full T-cell activation. This step is really important because it helps prevent our immune system from attacking our own body, keeping it in check and avoiding autoimmune reactions. Now, the activation of both humoral and cell-mediated immunity depends heavily on cytokines, which are signaling molecules produced by activated T helper cells. These cytokines help coordinate and amplify the immune response. It's also important to understand that T helper cells can only be activated when they encounter an antigen presented along with the right class II MHC molecules, plus a co-stimulatory signal. This tight regulation is key to ensuring that our immune system responds appropriately, not overreacting or attacking our own tissues. In summary, APCs are fundamental because they serve as the bridge between innate and adaptive immunity, ensuring our immune responses are well-regulated and targeted. Let's take a closer look at the essential properties of antigen-presenting cells, or APCs. They have two key features that are crucial for their role in immune activation. First, they express class II MHC molecules on their surface. These molecules are like display cases, holding antigenic peptides and presenting them to CD4-positive helper T cells, or Th cells. This presentation is vital because it helps the T cells recognize and respond to the specific pathogen or abnormal cell. The second important property is that APCs deliver co-stimulatory signals. They do this by expressing molecules such as B7, which includes CD80 and CD86. These molecules interact with CD28 on the helper T cells. This second signal is absolutely essential. Without it, the T cells won't become fully activated. Instead, they might become anergic or unresponsive. Once fully activated, these T cells can then proliferate and differentiate to carry out their immune functions. So, in summary, APCs are equipped with the tools to not only present antigens, but also provide the necessary signals to fully activate T cells, which keeps the immune response precise and effective. Early immunological studies revealed an important distinction between B cells and T cells. B cells recognize native, intact antigens, meaning they respond to the whole, unprocessed molecules. In contrast, T cells recognize processed antigen fragments, which are small pieces derived from the original antigen. Gell and Benacerraf demonstrated that secondary antibody responses depend on native antigen. When they denatured the antigen, making it lose its natural structure, the secondary antibody response was significantly reduced or even completely absent. This shows that B cells need the intact form of the antigen to respond effectively. However, their findings also indicated that T-cell responses are different. Secondary T-cell responses can be triggered by either native or denatured antigens. This suggests that T cells don't require the whole, intact antigen. Instead, they recognize specific fragments. Building on this, Szymonkewicz provided further proof by showing that peptide fragments of proteins can directly activate helper T cells. Interestingly, this activation occurs even when antigen-presenting cell membranes are fixed with formaldehyde, which essentially renders them inactive in terms of other functions. This experiment was crucial because it proved that T cells recognize short peptide fragments, not the entire protein. It highlights a fundamental difference in how B cells and T cells recognize antigens. B cells need the whole, native structure, while T cells focus on small processed pieces. Understanding these differences is key to grasping how the immune system distinguishes and responds to various pathogens and antigens. Let's start with Ziegler and Annanu's important contribution. They showed that T helper cell activation actually requires antigen processing. What does that mean? Well, when antigen-presenting cells, or APCs, were fixed before exposing them to the antigen, the T cells didn't get activated. But if the APCs were allowed to ingest the antigen and then fixed after 1 to 3 hours, T helper cells still got activated. This was a crucial finding because it demonstrated that antigen processing happens inside the APCs before the peptides are displayed on the surface. It highlights the essential step of internal processing before T-cell recognition. Moving on to cytotoxic T cells, Townsend and colleagues made a key discovery. They showed that these cells recognize short peptide fragments that come from internal viral proteins. They even demonstrated that synthetic peptides could be used to sensitize target cells for killing by cytotoxic T cells. This confirmed something very important. Both CD4 and CD8 T cells recognize processed peptides that are bound to MHC molecules. This process of peptide presentation is fundamental to how our immune system detects infected or abnormal cells, tying back to the overall theme of antigen recognition and immune activation. Now, let's look at the types of professional antigen-presenting cells, or APCs, and their key roles. These cells mainly activate previously sensitized T cells, known as T lymphocytes, which have already encountered their specific antigen and become primed to respond quickly upon re-exposure. This activation typically occurs at infection sites where the immune response is needed most. Focusing on the specialized cells, first, we have dendritic cells. These are considered the most efficient APCs because they are highly specialized for antigen uptake, processing, and presentation. They constantly express high levels of class II MHC molecules and co-stimulatory molecules, which are essential for activating naive helper T cells. This makes dendritic cells crucial in kickstarting immune responses from scratch. Next, we have macrophages. These cells also play a vital role, but in a slightly different way. Macrophages internalize antigens primarily through phagocytosis, engulfing pathogens and debris. They express class II MHC and B7 molecules, but only after they are activated. Once activated, macrophages can present antigens to T cells, helping to sustain and amplify the immune response. In summary, these APCs, dendritic cells and macrophages, are central to immune activation. Dendritic cells are particularly effective at initiating responses, especially by activating naive T cells, while macrophages are key players in ongoing immune responses, especially after they have been activated themselves. Let's start by looking at B lymphocytes, or B cells. These cells play a crucial role in our immune response. They internalize antigens primarily through a process called receptor-mediated endocytosis, which involves surface immunoglobulin receptors that specifically recognize and bind to antigens. Once they've captured an antigen, B cells process it and present pieces of it on their surface using class II MHC molecules. Interestingly, B cells always express these class II MHC molecules, even at rest, which allows them to present antigens to helper T cells. However, the co-stimulatory B7 molecules, which are necessary for full T cell activation, only appear after the B cell has been activated itself. This means B cells are mainly responsible for presenting antigens to helper T cells during antibody responses, helping coordinate a targeted immune attack. Now let's talk about non-professional antigen-presenting cells. These include cells like endothelial cells, epithelial cells, and fibroblasts. Under normal conditions, they don't usually present antigens, but during inflammation, they can be induced to express class II MHC molecules and co-stimulatory signals. This temporary expression enables them to participate in immune responses, but their role is limited and short-lived compared to professional APCs like B cells and macrophages. Finally, it's important to understand the different types of antigen-presenting cells, or APCs. Professional APCs are specialized for this task, but during certain inflammatory states, Non-professional APCs can also contribute. This dynamic ability of various cells to present antigens helps ensure our immune system can respond effectively in a variety of contexts. Let's talk about antigen processing first. This is the process where complex proteins inside the cell are broken down into smaller peptide fragments. It's like chopping up a large protein into tiny pieces that can be recognized by the immune system. Next, we have antigen presentation. This is when those peptide fragments are displayed on the surface of the cell, bound to MHC molecules. Think of it as the cell putting up a sign that shows what it has processed internally. It's important to remember that T cells, our immune sentinels, recognize only these peptide, MHC complexes. They do not recognize the native, whole antigens directly. This specificity is crucial for targeted immune responses. So, to connect everything, antigen processing and presentation are fundamental steps that prepare the immune system to detect and respond to pathogens. By breaking down proteins and displaying the fragments, cells effectively communicate their internal state, enabling T cells to identify threats accurately. Let's take a closer look at antigen processing and presenting. This process is crucial because it's how our immune system recognizes and responds to pathogens. First, when a pathogen enters the body, immune cells like macrophages and dendritic cells engulf it through a process called phagocytosis. Inside these cells, the pathogen is broken down into smaller fragments, or peptides. These peptides are then loaded onto major histocompatibility complex molecules, or MHC for short. There are two main classes, MHC class I and class II, and they each present these peptides to different types of T cells. MHC class I molecules present peptides from intracellular pathogens, like viruses, to cytotoxic T cells. Meanwhile, MHC class II molecules present extracellular pathogen peptides to helper T cells. This presentation is vital because it activates the T cells, which then coordinate a targeted immune response. Understanding this process highlights how our immune system can distinguish between normal cells and infected ones, allowing for precise and effective defense. So, in essence, antigen processing and presenting is a sophisticated way our immune system identifies threats and orchestrates a response. Let's take a closer look at the key features of the endogenous pathway for antigen presentation. This pathway processes intracellular antigens, such as viral proteins or tumor antigens, which are produced inside the cell. Importantly, this occurs in all nucleated cells, making it a widespread mechanism for immune surveillance. The peptides generated from these proteins are presented on the cell surface bound to MHC class I molecules. This presentation is crucial because it allows cytotoxic T cells, specifically CD8-positive TC cells, to recognize and respond to infected or abnormal cells. Now, let's walk through the steps of this process. First, endogenous proteins are degraded by proteasomes in the cytoplasm, producing peptide fragments. These peptides are then transported into the endoplasmic reticulum by TAP proteins. Inside the ER, the peptides bind to newly formed MHC class I molecules. Once the peptide, MHCI complex, is assembled, it moves to the cell surface. Here, the complex can be recognized by CD8-positive TC cells, which, upon recognition, can initiate the killing of the target cell. This pathway is fundamental for immune defense against viruses and tumor cells, highlighting the importance of intracellular antigen processing in immune surveillance. Now, let's explore how intracellular proteins are degraded into short peptides. This process is carried out by large protease complexes called proteasomes. Proteasomes are quite remarkable. They are large cylindrical structures, specifically 26S complexes, made up of multiple subunits that form a central catalytic channel. Proteins that are marked for degradation are often tagged with a small protein called ubiquitin. This ubiquitin tag acts like a signal, directing these proteins to the proteasomes for breakdown. Once inside the proteasome, the proteins are degraded in an ATP-dependent process, which means energy in the form of ATP is required. The result is the production of peptides that are of the right size for further processing. Now, some of these peptides are endogenous antigens, meaning they originate from proteins synthesized within the cell itself. This includes viral proteins if the cell is infected, tumor-associated proteins, or even abnormal self-proteins that shouldn't normally be present. These endogenous antigens are then processed through a specific pathway called the cytosylic pathway, which prepares them for presentation by class I MHC molecules. This pathway essentially uses the cell's existing protein degradation machinery, the same system involved in regular protein turnover. Finally, these processed peptides are presented on the cell surface to CD8-positive cytotoxic T lymphocytes, or TC cells. This presentation is crucial because it allows immune cells to recognize infected or abnormal cells, leading to targeted immune responses. In summary, the endogenous pathway for MHC class I presentation involves the degradation of intracellular proteins by proteasomes, processing of peptides, and their display on the cell surface to activate cytotoxic T cells, playing a vital role in immune surveillance. Now, when peptides are generated in the cytosol, they don't just stay there. They need to be transported into the rough endoplasmic reticulum, or RER, where they get loaded onto class I MHC molecules. This process is quite intricate and relies on the help of chaperone proteins like calreticulin and calnixin, which assist in stabilizing the peptide MHC complex during loading. The actual transport of these peptides is mediated by a specialized protein complex called TAP, or transporter associated with antigen processing. TAP functions as a heterodimer, meaning it's made up of two different subunits, TAP1 and TAP2, that work together. Importantly, TAP uses energy from ATP to move these peptides across the membrane into the RER. This ATP-dependent process is critical because it ensures that only properly processed peptides are loaded onto MHC class I molecules. Now, if there are defects in TAP, the transport process is impaired. This leads to reduced expression of MHC class I molecules on the cell surface, which can cause serious immune issues, including immunodeficiency or autoimmune-like disorders. So to tie it all together, this pathway, called the endogenous pathway, is essential for presenting intracellular antigens via MHC class I, which is a key part of our immune surveillance system. Now let's take a closer look at the key features of this pathway. It processes extracellular antigens, such as bacteria and soluble proteins, which are things our immune system needs to recognize and respond to. This process occurs specifically in professional antigen-presenting cells, or APCs, like dendritic cells, macrophages, and B cells. These cells are specialized for this task. The antigens are presented with MHC class II molecules, which are essential for alerting helper T cells, specifically CD4-positive T helper cells. When these T cells recognize the peptide MHC II complex, they get activated and help coordinate the immune response. Now let's go through the steps involved. First, antigens are taken up by endocytosis or phagocytosis. That's how the cell engulfs these foreign particles. Once inside, the antigens are degraded inside endolysosomes, breaking them down into smaller peptide fragments. These peptides then bind to MHC class II molecules inside vesicles. After binding, the peptide, MHC II complex is transported to the cell surface, where it is displayed. This display is crucial because it allows helper T cells to recognize the antigen. When a T helper cell recognizes this complex, it becomes activated and releases cytokines that help orchestrate the immune response. This entire process is part of what we call the exogenous pathway, or the class II MHC pathway. It's specifically tailored for extracellular antigens, helping our immune system identify and respond effectively to pathogens outside our cells. Recognizing these features and steps is key to understanding how our immune system detects and reacts to threats in our environment. Exogenous antigens are essentially extracellular antigens that enter antigen-presenting cells, or APCs, from outside the cell. Once inside, these antigens are processed via the endocytic pathway. This means they're taken up through mechanisms like phagocytosis or endocytosis, and then broken down into smaller peptide fragments. These fragments are then loaded onto class II MHC molecules, which are specialized for presenting extracellular antigens. The peptide class II MHC complex is recognized by CD4 positive T helper cells, which are crucial for coordinating immune responses. It's important to note that this pathway differs from the endogenous or cytosolic pathway, mainly in how the antigens enter the cell, where they're processed, and the type of MHC molecules involved. Exogenous antigens are internalized by APCs through several methods. Phagocytosis, mainly performed by macrophages and dendritic cells, is one key route. Then there's endocytosis, which includes receptor-mediated endocytosis, particularly in B cells, which use antigen-specific surface immunoglobulins, and pinocytosis, often called cell drinking. Macrophages are versatile. They can internalize antigens through both phagocytosis and endocytosis. B cells, on the other hand, mainly rely on receptor-mediated endocytosis because they use their surface immunoglobulins to specifically capture antigens. This entire process is central to how the immune system recognizes and responds to extracellular threats, linking the physical internalization of antigens to the activation of CD4 T cells and the broader immune response. Let's talk about how internalized antigens are processed inside the cell. Once they are taken up, they are degraded into peptides within acidic endocytic compartments. The pathway they follow includes three main, progressively more acidic compartments. First are the early endosomes, which have a pH around 6 to 6.5. Then come the late endosomes or endolysosomes, with a pH between 5 and 6. Finally, we have the lysosomes, which are even more acidic, with a pH roughly 4.5 to 5. Lysosomes are packed with over 40 different acid-dependent hydrolytic enzymes. These include proteases, nucleases, lipases, and glycosidases. Their job is to break down the antigens into smaller fragments, specifically oligopeptides of about 13 to 18 amino acids. These sizes are ideal because they fit well into the binding groove of class II MHC molecules. It's also important to note that antigen processing can be blocked or inhibited. For example, agents that raise the pH inside these vesicles, like chloroquine, can interfere with the degradation process. Similarly, protease inhibitors like lupeptin can prevent the enzymes from breaking down the antigens effectively. This entire process is crucial in the exogenous pathway, which is the class II MHC pathway. It allows the immune system to present external antigens to helper T cells, enabling an appropriate immune response. Understanding this pathway helps us appreciate how the immune system detects and responds to foreign invaders. Class II MHC molecules are initially synthesized in the rough endoplasmic reticulum, or RER, where they begin their journey. Once formed, they associate with a special trimeric protein called the invariant chain, also known as I or CD74. This invariant chain has two critical roles. First, it blocks the peptide binding groove of the MHC molecule, which is important because it prevents endogenous or self-derived peptides from binding at this stage. Second, it acts as a guide, directing these MHC invariant chain complexes toward endocytic compartments within the cell. The sorting signals embedded in the cytoplasmic tail of the invariant chain are key to this process. They essentially serve as a GPS, guiding the entire complex from the Golgi apparatus into endosomes. This pathway is central to the exogenous pathway of antigen presentation, where external or extracellular antigens are processed and presented by MHC class II molecules. Connecting this to the bigger picture, this pathway allows immune cells to effectively sample and respond to pathogens that invade from outside the cell, which is essential for initiating a proper immune response. Let's take a closer look at the pathway. Here, I want to emphasize how essential it is to understand the steps involved in progressing forward. This pathway represents not just a series of actions, but a strategic route that guides us toward our goals. As we move along this route, each key point connects to the overall theme of growth and improvement. It's important to recognize that the pathway is designed to be clear and purposeful, helping us stay focused and aligned with our objectives. By following this pathway, we can ensure that our efforts are coordinated and effective, ultimately leading us to success. Let's take a closer look at the pathway. This slide highlights the key steps and processes involved in achieving our goals. First, we start with identifying the initial step, which is understanding the current situation and setting clear objectives. From there, we move into planning, where we develop strategies and outline the actions needed. The next phase involves execution, where these plans are put into action with focus and precision. Throughout this process, monitoring and evaluation are essential to ensure we're on track and making necessary adjustments. Ultimately, this pathway connects all the dots, showing how each phase supports our overall mission to improve and innovate continuously. It's a clear roadmap that guides us forward, emphasizing the importance of a structured yet adaptable approach. Now, let's look at the key functions of APCs in immune response. First, they initiate the adaptive immune response by activating naive T helper cells. This activation is crucial because it sets the stage for both humoral or antibody-mediated responses and cell-mediated defenses. Next, APCs play a vital role in regulating immune responses. They do this through controlled antigen presentation and co-stimulation. This regulation is essential to prevent the immune system from attacking our own tissues, which would lead to autoimmunity. Moving on, APCs produce cytokines that are like messengers, influencing how T cells differentiate and what kind of immune response develops. These cytokines help direct the immune system to respond appropriately to different threats. Finally, once the helper T cells are activated by APCs, they assist in activating cytotoxic T cells and B cells. This cooperation enables the production of antibodies and the killing of infected or abnormal cells, completing the immune defense cycle. So, in summary, APCs are central to launching and regulating our immune responses, ensuring they are effective but also controlled to prevent harm to our own body. Nearly all nucleated cells express class I MHC molecules, which are essential for immune surveillance. These molecules present endogenous antigen fragments, those originating from within the cell, to cytotoxic T cells. Essentially, any nucleated cell can present these internal peptides, allowing the immune system to monitor cell health and identify abnormal or infected cells. Target cells that are commonly recognized include virus-infected cells, tumor cells, and cells that are aged or damaged. Additionally, transplanted allogeneic cells can also be targeted if recognized as foreign. When we look at the characteristics of these target cells, it's important to understand that their ability to present antigens via MHC class I molecules is what enables cytotoxic T cells to identify and eliminate cells that are abnormal or compromised. This process is a critical part of our immune defense, especially in protecting against infections and cancer, and in rejecting transplanted tissues. Let's take a closer look at class I versus class II MHC presentation. Class I MHC molecules are found on almost all nucleated cells, which means they have a broad presence throughout the body. Their main role is to present endogenous antigens, those originating inside the cell to CD8-positive cytotoxic T cells. This is crucial for identifying and destroying infected or abnormal cells. On the other hand, class II MHC molecules are more specialized. They are expressed only on professional antigen-presenting cells, or APCs, like dendritic cells, macrophages, and B cells. These molecules present exogenous antigens, coming from outside the cell to CD4-positive helper T cells. This interaction is vital for orchestrating a coordinated immune response. Now, let's clarify the difference between target cells and antigen-presenting cells. Cells that display antigens via class I MHC to CD8 T cells are called target cells. They are usually the ones being attacked or surveilled. Cells that present antigens with class II, MHC, to CD4 T cells are called antigen presenting cells, or APCs. These are the cells that initiate and regulate the immune response. Finally, understanding MHC restriction is key. It's about which T cells recognize which MHC molecules, and it explains why only certain T cells respond to specific antigens presented by particular cells. This concept links directly to the target cells of APCs, emphasizing how the immune system is finely tuned to distinguish between different types of threats and cells. Let's take a closer look at CD1 molecules. These are non-classical MHC class I-like molecules, which means they are similar to the classical MHC molecules, but serve a slightly different function. Their main role is to present lipid and glycolipid antigens to T cells, especially to a special subset known as natural killer T, or NKT cells. Now, unlike the classical MHC molecules that are known for presenting peptide antigens, CD1 molecules focus on lipid-based antigens. This is particularly important because many pathogens, like mycobacteria, have cell walls rich in lipids. These lipids can be recognized by the immune system through CD1 molecules, which makes them crucial in fighting certain infections. Moving on to the pathway itself, the CD1 antigen presentation pathway involves the processing of lipid antigens within cells, and then their presentation on the surface via CD1 molecules. This pathway is specialized, allowing the immune system to detect and respond to lipid-based components of pathogens that might otherwise go unnoticed by traditional peptide presentation pathways. Overall, understanding how CD1 molecules differ from classical MHC molecules helps us appreciate the diversity and adaptability of our immune system in recognizing a wide array of pathogen components. This highlights the importance of lipid antigen presentation in our immune defense. Now, let's look at how different CD1 molecules have unique intracellular localization patterns, which suggests that they use specialized pathways for antigen processing and presentation. Focusing on CD1, these molecules are mainly found on the cell surface and within recycling endosomes. Recycling endosomes are key compartments involved in internalizing extracellular material and representing it to the immune system. This strategic localization allows CD1 to sample lipid antigens that are present at the cell surface and inside early endocytic compartments. As a result, CD1 primarily presents extracellular lipid antigens that are taken up through endocytosis. This pathway is particularly important because it enables rapid antigen presentation, leading to early immune activation. So overall, the localization of CD1 supports its role in quickly identifying and presenting extracellular lipids to initiate an immune response. Let's take a closer look at CD1b and CD1d and where they are located within the cell. These molecules are primarily found within late endosomes and lysosomal compartments. Now, lysosomes are really important organelles involved in breaking down and processing complex antigens. They contain an acidic environment and a variety of digestive enzymes that work together to efficiently break down lipids and other molecules. This environment is crucial because CD1b and CD1d present processed lipid and glycolipid antigens that come from intracellular pathogens. So, their positioning within these compartments allows them to access and load these lipid antigens effectively. This is especially significant when it comes to pathogens like mycobacteria, which have cell wall components rich in lipids. The localization of CD1b and CD1d in these compartments is key for processing these lipid-rich structures and presenting them to T cells. In essence, the pathway of antigen presentation by CD1 molecules hinges on this strategic localization within the cell, enabling the immune system to recognize and respond to lipid-based components of intracellular pathogens efficiently. Cytokines are small, soluble proteins with low molecular weight that are secreted by immune cells, and interestingly, some non-immune cells as well. Their main role is to regulate and coordinate immune responses, inflammation, and even hematopoiesis, which is the formation of blood cells. Think of cytokines as messengers, intercellular signaling molecules that transmit signals between cells. This signaling controls various processes like cell growth, differentiation, activation, and overall function, both in immune and non-immune cells. They are absolutely central to our immune system, playing a crucial role in both innate and adaptive immunity. They help control how immune cells communicate, activate, and regulate each other, ensuring a balanced and effective immune response. So, in essence, cytokines are vital for maintaining immune system harmony and responding appropriately to threats. Let's take a closer look at the general characteristics of cytokines. These are glycoproteins or polypeptides, typically with low molecular weights ranging from about 8 to 30 kilodaltons. They are produced transiently and locally, usually in response to immune stimuli like infection, injury, or inflammation. This means they are quickly made and act close to where they are produced, ensuring a rapid and targeted immune response. Cytokines bind to specific high-affinity receptors on target cells, which allows them to exert precise biological effects. Even at very low concentrations, often in the picomolar or nanomolar range, they can trigger powerful biological responses. This highlights how potent they are despite their small amounts. One key feature of cytokines is their pleiotropy, meaning a single cytokine can act on different cell types and produce various effects. For example, one cytokine might influence immune cells, tissue cells, or other cell types, depending on the context. Additionally, cytokines exhibit redundancy, where different cytokines can perform similar functions, providing a backup system to ensure immune regulation. Cytokines don't work in isolation. They often operate within complex networks and cascades. One cytokine can induce the production of others, amplifying or modulating the immune response. Their mode of action can also vary, acting autocrinely, meaning on the same cell that produces them, paracrinely on neighboring cells, or endocrinely, affecting distant cells through the bloodstream. All of these features, their potency, versatility, and interconnected functions, highlight the central role cytokines play in immune regulation and response. Let's take a closer look at the sources of cytokines. Cytokines are produced by a wide variety of cells in our body, each playing a specific role. For example, T lymphocytes, or T cells, produce cytokines that are crucial for regulating our adaptive immune responses. These are the cells that help tailor our immune defense to specific pathogens. B lymphocytes, or B cells, also produce cytokines, but their main role is in antibody production, which is essential for targeting and neutralizing pathogens. Moving on, macrophages and monocytes are key players in our innate immune system. They produce cytokines involved in inflammation and are the first responders to infection. Dendritic cells are another important source. They produce cytokines that help regulate antigen presentation and activate T cells, effectively bridging innate and adaptive immunity. Then we have natural killer cells, or NK cells, which secrete cytokines that are vital in fighting off viruses and tumors, providing an immediate immune response. Finally, endothelial cells and fibroblasts, which are involved in tissue repair, produce cytokines during inflammation and healing processes. So, as you can see, cytokines come from many different cell types, each contributing to the complex orchestration of immune responses and tissue maintenance. Understanding these sources helps us appreciate how interconnected and dynamic our immune system truly is. Now let's look at how cytokines are classified into several major groups. First, we have the interleukins, or ILs. These molecules are crucial for communication between leukocytes, or white blood cells, and they play a vital role in controlling immune cell growth and differentiation. Some common examples include IL-1, IL-2, IL-4, IL-6, IL-10, and IL-12. Each of these has specific functions in modulating immune responses. Next, we have the interferons, or IFNs. These cytokines are especially important because they possess antiviral, immunoregulatory, and antiproliferative activities. For instance, IFN-alpha, IFN-beta, and IFN-gamma are well-known examples. They help coordinate the immune response against viruses and also regulate cell growth and immune activation. Understanding these classifications helps us appreciate how cytokines orchestrate the immune system's complex responses, which is a key theme in our overall discussion of immune regulation and defense mechanisms. Now, let's move on to some other important cytokines. First, we have tumor necrosis factors, or TNFs. These molecules play a crucial role in regulating inflammation, controlling apoptosis, and activating immune cells. Essentially, they help coordinate the body's response to injury or infection. Two common examples are TNF-alpha and TNF-beta, which are key players in inflammatory processes. Next, we have colony-stimulating factors, or CSFs. These are vital for blood cell production, as they stimulate the bone marrow to produce and differentiate various blood cell types. For instance, GM-CSF encourages the growth of multiple white blood cell lineages, while G-CSF specifically promotes neutrophil production, and M-CSF supports macrophage development. These factors are essential for maintaining healthy immune function. Finally, let's discuss chemokines. These are specialized cytokines that direct cell migration and trafficking, especially during immune responses and inflammation. They essentially act as the body's GPS, guiding immune cells to sites where they're needed most. Examples include also known as CXCL8, which attracts neutrophils, MCP1, which recruits monocytes, and RANTES, involved in attracting various immune cells. Understanding these chemokines helps us grasp how the immune system orchestrates a coordinated response. Now, let's take a closer look at the different modes of action of cytokines. First, in autocrine action, cytokines act on the very same cell that secreted them. This means the cell basically regulates its own activity and functions through these signals. A good example of this is interleukin-2, or IL-2, which acts on activated T cells to stimulate their proliferation and differentiation. So, the cell is essentially calling its own shots to grow and develop. Next, we have paracrine action. In this case, cytokines influence neighboring cells, which helps modulate local immune responses. Think of cytokines like signals that coordinate activities in the immediate environment. For example, interleukin-1, or IL-1, acts on nearby endothelial cells and leukocytes to induce inflammation. This localized response is critical for containing infections and initiating healing processes. Finally, there's endocrine action. Here, cytokines enter the bloodstream and travel to distant organs, producing systemic effects. It's like a hormonal message traveling through the body. A prime example is tumor necrosis factor alpha, or TNF-alpha, which can cause fever and stimulate the liver to produce acute phase proteins. This illustrates how cytokines can have widespread impacts beyond the local environment. In summary, these three modes, autocrine, paracrine, and endocrine, highlight the versatile ways cytokines regulate immune responses, from local actions to systemic effects. Understanding these mechanisms helps us grasp how the immune system coordinates complex responses to threats. Now, let's delve into the functions of cytokines in more detail. First, cytokines play a crucial role in regulating the growth and differentiation of immune cells. This means they control how T cells, B cells, macrophages, and other leukocytes develop, ensuring that the immune system can respond appropriately to threats. Next, cytokines are vital for activating immune responses. They stimulate immune cells and boost their effector functions, which essentially enhances the body's ability to eliminate pathogens and abnormal cells effectively. Finally, cytokines are key mediators of inflammation. They help coordinate both acute and chronic inflammatory responses by causing vasodilation, increasing vascular permeability, and recruiting leukocytes to the site of infection or injury. Understanding these functions highlights how cytokines are central to orchestrating an effective immune response, connecting all these points to the broader theme of immune regulation and defense. Let's take a closer look at how cytokines contribute to our immune defense. First, in the realm of antiviral defense, interferons play a crucial role. They inhibit viral replication inside host cells, which not only helps protect those infected cells, but also creates a shield for neighboring cells, preventing the virus from spreading further. This process is essential for controlling viral infections early on. Next, we have the regulation of hematopoiesis. Colony-stimulating factors are key players here. They stimulate the production and differentiation of blood cells in the bone marrow. This regulation is vital because it ensures our blood cell levels stay balanced, supporting overall immune function and recovery after infections or injuries. Finally, chemokines are like the body's GPS for immune cells. They guide immune cells to the exact locations where they're needed, be it infection sites, areas of inflammation, or tissue injury. This directed migration not only enhances immune surveillance, but also ensures that our immune response is both targeted and efficient. All these functions, antiviral defense, blood cell regulation, and cell migration, highlight how cytokines orchestrate a coordinated and effective immune response, which ties back into the overall theme of understanding immune system regulation and defense mechanisms. Let's take a closer look at the role of cytokines in different physiological responses. First, we have the induction of fever and the acute phase response. Cytokines such as IL-1, IL-6, and TNF-alpha play a crucial role here. They act as messengers that signal the brain to raise the body temperature, which is a key part of our defense mechanism against infection. At the same time, these cytokines stimulate the liver to produce acute phase proteins, which help combat pathogens and facilitate healing. This coordinated response enhances our ability to fight off invaders. Next, we move to the regulation of apoptosis. Some cytokines are essential in controlling programmed cell death. This process is vital for maintaining immune system homeostasis, ensuring that harmful or excess cells are eliminated efficiently. By regulating apoptosis, cytokines help prevent autoimmune reactions and keep our immune responses balanced. Finally, cytokines are deeply involved in wound healing and tissue repair. They stimulate fibroblast proliferation, which is necessary for rebuilding tissue. They also promote collagen synthesis, providing structural support, and encourage new blood vessel formation, known as angiogenesis, to supply nutrients and oxygen to the healing tissue. All these actions work together to promote effective tissue repair and wound healing, demonstrating just how versatile and vital cytokines are in maintaining our health and recovery processes.